Human Papillomavirus and Oral Cancer by Julie R. Jennings. Significance of exposure. 1 in 4 Americans are infected with human papillomavirus, or HPV. It is estimated that there are nearly 20 million new sexually transmitted diseases in the United States annually, and HPV attains for nearly 50% of these newly acquired sexually transmitted infections. It is expected that by the year 2020, HPV-related oropharyngeal cancer, or OPC, will have a higher incident rate than cervical cancer. The cost of treatment for OPC is a concern for healthcare providers. Americans spend between $160 million and $1.6 billion on non-cervical HPV-related disease. Other burdens of treatment include psychological issues and quality of life issues. Significance of outcome. Head and neck squamous cell carcinoma, or HNSCC, is the 6th leading cause of cancer worldwide. It is estimated that in the year 2016, there will be 61,760 new cases of HNSCC in the United States. About 70% of OPCs are caused by the HPV virus. The 5-year survival rate of OPC varies upon stage, number of lymph nodes involved, HPV status, and history of smoking. The 5-year survival rate for HPV-positive OPC is 70 to 80%. HPV-positive HNSCC has a 54% better overall survival compared to HPV-negative patients. To find the articles used in the analysis, PubMed and Sinhal were searched for relative current research related to HPV and OPC. 3 primary study articles were analyzed to determine if HPV status is directly related to the development of OPC. Articles were limited to those that were English articles published in the last 10 years. The articles each contained statistical evidence that looked at the relationship between HPV and OPC. Case number 1, case control study of human papillomavirus and oropharyngeal cancer. The case sample consisted of 100 participants that were newly diagnosed with oropharyngeal cancer between the years 2000 and 2005. The participants were outpatients at the otolaryngology clinic of the Johns Hopkins Hospital in Baltimore. The control sample consisted of 200 participants without cancer who were seen for a benign condition at the otolaryngology clinic of the Johns Hopkins Hospital in Baltimore from 2000 to 2005. Eligible control participants were the same sex and a 5-year age category were approached until 2 controls were matched to each participant. The data collected was oral mucosal specimen, serum specimen, and tumor sample. The samples of the case patients were obtained before any therapy was done. Additional variables reported include demographic characteristics, oral hygiene, medical history, family history of cancer, lifetime sexual behaviors, lifetime history use of marijuana, tobacco, and alcohol. Strengths of the study were that HPV status was identified using multiple methods. Weaknesses of the study include a small sample size and the case control study design. In case control studies, exposure and outcome are assessed at the same time, therefore it is uncertain if the exposure precedes the outcome. Study number 2. Demographics and risk factors in patients with head and neck tumors. There were 86 case participants who were treated between the years 2000 and 2004 at the Department of Otolaryngology Head and Neck Surgery in Prague. The control participants were also treated between the years 2000 and 2004 at the Department of Otolaryngology Head and Neck Surgery, but did not have oncological diagnoses. Data was collected using oral exfoliant cells, tumor specimens, and serological assay for presence of antibodies. Strengths of the studies included multiple methods of identifying HPV status, Weaknesses include a small sample size and the use of a case control study design. Study number 3, human papillomavirus infection and oral cancer, a case control study in Montreal, Canada. The 72 case participants were newly diagnosed with squamous cell carcinoma of the mouth, palatine tonsils, and oral pharynx. The participants were patients at one of 3 head and neck surgery services in Montreal between October 1997 and May 2001. The control participants were 129 patients from the same hospitals where cases were recruited. The control participants were selected using a frequency match on the basis of gender, age, and hospital between 1997 and 2001. Data was collected using oral exfoliated cells for HPV DNA and serological response to the virus. Strengths of the study include assessing HPV status with 2 markers and determining control participants by matching them with case participants. Weaknesses include a small sample size and the use of a case control study design. Here's a table to compare the information previously given about each article. Results of study number 1. The results show that 37% of case participants were HPV positive, while only 6% of the control participants were HPV positive. The adjusted odds ratio was 14.6, with a 95% confidence interval of 6.3 to 36.6. These results show that this data is statistically significant and that there is a strong association between HPV and oral cancer. Results of study number 2. The results show that 48.8% of case participants were HPV positive, while only 22.7% of control participants were HPV positive. The adjusted odds ratio was 10.7 and the 95% confidence interval was 4.7 to 24.7. These results show that this data is statistically significant and that there is a strong association between HPV and oral cancer. Results of study number 3. The results show that 19.4% of case participants were HPV positive, while only 4.7% of control patients were HPV positive. The adjusted odds ratio was 3.14 and the 95% confidence interval was 0.9 to 10.9. These results show that there is a possibility that the results were due to chance. Therefore, the results are not statistically significant. Heniken's criteria. As stated previously, studies 1 and 2 show statistical significance, while study 3 was not statistically significant. The risk for bias in all 3 studies include selection bias and confounding bias. The confounding variables include age, gender, family history, alcohol use, tobacco use, oral hygiene, health history, family history of cancer, race, religion, and language. The biological plausibility refers to whether or not there is a logical proposed possible mechanism for the cause and effect relationship. Each of the articles had this. Consistency refers to a pattern of similar results found by different investigators at different times. Study 3 was the only article that did not show consistency. Temporality refers to whether the disease occurred after the exposure. This is not determined in any of the studies because they used a case control design and the participants already had the disease at the beginning of the study. Dose response refers to a relationship in which a greater exposure to the pathogen results in greater risk of contracting the disease, which none of the articles had this. Conclusion. All of the studies showed moderate to strong association between HPV and OPC. One of the studies did indicate a confidence interval that showed a possibility of chance related to the association of HPV and OPC. Overall, there's enough evidence from the studies to show that there's an association between HPV and OPC. Further research. There are currently multiple vaccines available to prevent high-risk HPV types. Further research should look at relationships between OPC and HPV vaccination status and relationship between HPV status and HPV vaccination status.